Roles of diagnostic cytology. Fast forward. Cytology is the study of cells, including their structure and function. It involves examining cells under a microscope to assess their morphology and characteristics. So cytology is widely used in medical diagnostics, research, and forensic investigations. Whereas cytopathology is a branch of pathology that focuses on diagnosing diseases at cellular level. It involves studying cells obtained from various body sites to detect abnormalities, infections, and cancers. It is commonly used in screening, diagnosis, and monitoring of diseases. So one of the roles of diagnostic cytology is early detection of diseases. For example, cancers, where pap smear is used to detect precancerous cells in the cervix, allowing timely intervention. So precancerous cells are abnormal cells that have undergone genetic or structural changes, increasing their potential to develop into cancer if left untreated. These cells show uncontrolled growth and are not yet cancerous but remain confined to their original location. So some of the genetic changes that can lead to precancerous transformation include activation of proto-oncogenes. Proto-oncogenes are normal genes that regulate cell growth. When mutated, they become oncogenes. So oncogenes are mutated versions of normal genes that regulate cell growth. So when activated, they cause uncontrolled cell proliferation, driving excessive proliferation. In precancerous cells, low-level oncogene activation occurs, leading to increased but still controlled growth. Examples of proto-oncogenes, we have the rat sarcoma genes, which encode proteins that regulate cell growth, differentiation, and survival by transmitting signals from cell surface receptors to the nucleus. Mutation of RAS genes promote excessive but non-invasive cell proliferation in early lesions. Another example, myelocytomatosis oncogene, which is a transcription factor that regulates genes involved in cell proliferation, metabolism, apoptosis, and differentiation. Overexpression of this gene increases cell division in early stage dysplastic tissue. Number 2, inactivation of tumor suppressor genes. Tumor suppressor genes regulate cell division and repair damaged DNA. Their loss removes growth restraints, allowing uncontrolled division. So the tumor suppressor gene act as brakes on cell division. Their partial loss or silencing allows precancerous cells to proliferate, though they have not yet invaded. In early stages, cells may have one inactivated copy of a tumor suppressor gene, what we call haploinsufficiency, but a second hit or mutation is needed for full malignancy. Examples: adenomatous polyposis coli, which is a mutation found in colon polyps, a precancerous lesions in familial adenomatous polyposis. Another example, retinoblastoma 1, which is a tumor suppressor gene that regulates the cell cycle by controlling the G1 and synthesis checkpoints. So partial inactivation leads to excessive cell cycle progression in early dysplasia. Number 3, DNA repair defects. So defects in DNA repair mechanisms cause an accumulation of genetic mutations, increasing the risk of cancer development. In precancerous cells,
Mild defects in repair genes may allow mutations to accumulate slowly. Examples: mismatch repair gene defects, which leads to microsatellite instability, a hallmark of colorectal adenomas. Another example, breast cancer gene 1 and 2, which are tumor suppressor genes involved in DNA repair through the homologous recombination repair pathway. Mutations can cause increased DNA damage. However, they require additional mutations for cancer development. Number 4, telomere shortening and early senescence. So normal cells have telomeres, which are protective ends of chromosomes that shorten with each division, eventually triggering cell death. Cancer cells reactivate telomerase, an enzyme that rebuilds telomeres, making them effectively immortal. In precancerous cells, telomeres are critically short, leading to genomic instability, but telomerase is not yet fully activated, so the cells have limited lifespan. Example, short telomeres in Barrett's esophagus that increases the likelihood of mutations leading to esophageal adenocarcinoma. Number 5, epigenetic changes. Epigenetic changes are heritable modifications in gene expression that do not alter the DNA sequence itself but affect how genes are turned on or off. These genes regulate various biological processes, for example, development, differentiation, and disease progression, for example, in cancer. Epigenetic modification can silence tumor suppressor genes. Classical examples or types of epigenetic changes. One, we have DNA methylation, addition of methyl groups to cytosine bases that leads to gene silencing by preventing transcription factor binding. Number 2, histone modifications. Histones are proteins that help package DNA into chromatin. Therefore, histone acetylation, which is a chemical modification, loosens chromatin and promotes gene activation. And histone methylation can activate or repress gene depending on the site. DNA methylation and histone modifications alter gene function in precancerous cells. For example, P16 gene is a tumor suppressor gene that regulates the cell cycle. Hypermethylation of P16 gene increases uncontrolled cell division in cervical dysplasia. 6. Increased resistance to apoptosis. Precancerous cells begin acquiring anti-apoptotic features. In other words, they avoid programmed cell death, but they are not yet completely resistant to cell death like full-blown cancer cells. Example, mild expression of B-cell lymphoma 2. This is an anti-apoptotic protein that plays a critical role in cell survival by inhibiting programmed cell death. Mild overexpression of this protein prevents apoptosis in early dysplastic cells, like in follicular lymphoma precursors. Another example is P53 protein, which is a tumor suppressor protein that regulates the DNA damage response, apoptosis, and cell cycle arrest. So P53 mutations leads to defective DNA damage response in early precancerous lesions. 7. Early angiogenesis. In some precancerous lesions, cells begin producing vesicular endothelial growth factor to promote blood vessel formation, but they do not yet invade blood vessels. Example is vesicular endothelial growth factor. that is a key signaling protein involved in angiogenesis. It plays a crucial role in both normal physiology and tumor development. Increased expression of this protein in high-grade cervical dysplasia supports cell survival but is not yet cancerous. 8. Early loss of adhesion and cell polarity. In other words, pre-metastatic changes begin. Cells begin to lose adhesion molecules like E-cadherin, making them loosely connected but still confined. So E-cadherin is a calcium-dependent cell adhesion molecule that plays a very important role in maintaining epithelial cell integrity and suppressing tumor invasion. So the E-cadherin forms adherin junctions connecting epithelial cells, maintaining tissue architecture and preventing cell detachment. Polarity loss is a sign of high-grade dysplasia. Example, partial loss of E-cadherin in ductal carcinoma in situ marks a transition towards invasive breast cancer. Examples of precancerous conditions: cervical dysplasia or the cervical intraepithelial neoplasia found in pap smear screenings and linked to human papillomavirus infection. This can progress to cervical cancer if untreated. Number 2, colon polyps, the adenomas, which are precancerous growths in the colon or rectum. So some types of adenomatous polyps can develop into colorectal cancer. Number 3, atypical ductal hyperplasia or lobular hyperplasia, abnormal cell growth in the breast ducts or lobules that increases the risk of breast cancer. Characteristics of precancerous cells. Number 1, cell atypia. In other words, abnormal cell appearance. So cells show irregular shapes and sizes, what we categorize as pleomorphism. Also, increased nuclear size relative to cytoplasm. In other words, we are having high nuclear to cytoplasmic ratio. Cells also experience hyperchromatic nuclei, darkly stained due to excess DNA. Number 2, disorganized cell growth, dysplasia, whereby there is loss of normal cell arrangement and organization. Cells do not align properly with their neighbors, loss of polarity, and may be graded as mild, moderate, or severe dysplasia based on severity. 3, increased and abnormal cell division. Cells have higher mitotic activity than normal cells, and there is presence of abnormal mitotic figures, irregular spindle formations. 4, loss of normal cell function. So precancerous cells lose their specialized function. In other words, they do not behave like their normal tissue counterparts. 5, genomic instability and DNA mutations. Mutations in genes controlling cell growth, such as tumor suppressor genes like p53, and activation of oncogenes that promote uncontrolled proliferation. Increased proliferation without invasions. So cells divide excessively but remain confined within their original tissue, with no invasion of the basement membrane. And number 8, angiogenesis. So in some cases, some precancerous cells start stimulating new blood vessel formation to support their growth.